It's about engineering the cells outside the lab. You pick the cells from the body of the recipient or the donor, whatever. There are various models. Let's focus on the recipient part, the one who's going to give the cells. So we pick up the cells, we take it to the lab, we engineer them, we make them slightly a bit more intelligent. That's what it is all about. Now, this is one part. How do we do it? Let me come to the second bullet. It's all Boolean logic. Or we can say binary. I believe everybody have had the taste of binaries during our childhood days. It's 1 and 0, right? So we program the cells. It will sound a bit like a computer or tech savvy or like a tech savvy part. But let me tell you, this is how it works. It works in the Boolean logic. Either one. Suppose I infuse a cell, one particular cell. Forget about CAR-Ts and all that. CAR-Ts, armored T cells, and all that. I'll come to that later. So I infuse a cell. The cell will see the cancer cell, find it out. The signal will be 1. So it'll go and hit the cancer cell. And if the signal is 0, it's not going to hit. So now we are using this Boolean logic to trap those cells, those notorious cancer cells, which are there in the body. And at the same time, there are various models out there. You can see it's CD83, CD33, 123, CLL1. I'll talk about this, all of them. But what we need to do is that at the end of the day, the take-home message would be put much pressure on the GVL part, as I said on the first slide. Graft versus leukemia reaction. And I told you, cancer cells are very smart. I'll prove it to you then, in that case. Okay, let's first talk about CRISPR. It's called CRISPR-Cas9 RNA complex. That RNA is a guide RNA. Let me speak 1 or 2 lines about this. You see, how does CRISPR work? Let's forget about malignancy for the time being, or neoplasia for the time being, or dysplasia for the time being. Let's forget about all these parts. Let's just think, how does CRISPR work? Cas9, I said CRISPR, Cas9 and guide RNA. These are the 2 things where you need to focus. Cas9. What is Cas9? Cas9 is just a scissor. It's a scissor to cut, right? And guide RNAs are like hairpins, which women usually use. So what happens? Suppose for an infection, say I take an infective subset, what we do is that we edit the entire genome, or we have a predelection or a foci to edit the entire genomic sequence, or to make genomic edits. So in that case, what we do is that we find out, in case of infections, I said it, a palindromic sequence where the sense and the antisense codon is going to read in the same way, like ATG, and from the other side, from 5' to 3', it is going to be ATG, and from 3' to 5', the antisense codon is going to be ATG, it's going to be GTA. So there we place the hairpin, and that's the guide. That's the guide particularly. That's going to guide me to that culprit genome, or the genomic sequence. And then we put the Cas9 out there, and the Cas9 cuts out the entire part, the entire troublesome part. Same for cancer. Almost the same, rather. We detect the part which is faulty, and we cut them off straight away. Right? So this is the CRISPR method, which we use during ex vivo engineering. Ex vivo, right? All right. I don't think so. I don't think so. I need to add much more out here. Let's go to the next slide. Right. So what scientists started to think? They started to think that GVHD, that is the target, right? Graft versus host disease. How we should mitigate that part and how we should accentuate GVL, graft versus leukemia reaction. That's what scientists were thinking. And what they have done is they have broken this part in two components. They started to think them as two different components. दोनों को पहले एक करके चल रहा था, अभी नहीं है वो. अभी ऐसा है कि आप एक को एक करो, you differ, you make a, you know what, you actually think this GVHD and GVL as two separate entities in which we need to reduce GVHD to increase GVL. That's what scientists are thinking. You see the source, it's there in front of us, right? It's a very high-end and new topic. Well, as far as CRISPR was concerned, I forgot to tell you that CRISPR has the ability to cut 20 genomic sequences, genomic edits, around 20. Some say it was 9. Then it got accentuated to 2, 13, I believe, 9 to 13, and now it can cut up to 20. So that's the power of CRISPR-Cas9 RNA. There. Right. So from here the model starts. See, when we engineer CAR cells, this I have mentioned two interleukins, 12 and 18. So what do? A cell will of course produce cytokines and stuff. Let's just focus on IL-2. IL-12, interleukin 12 and interleukin 18. What are they going to do? They're gonna go inside the body of the patient and they are gonna start producing a hell amount of, they're gonna recruit rather, like kings, they're gonna recruit a hell lot of police forces, which I call T cells. Number one, they're gonna recruit macrophages, they're gonna recruit natural killer cells, and they're gonna fight. For IL-18, interleukin 18, what do? What happens actually? For interleukin 18, the biggest part being, it has got a very strong, it anchors the entire scenario. Let's just take it in this way. Both are, friends. They love working together. IL-12, interleukin 12 and IL-18, they love working together. But interleukin 18 actually anchors the entire part. That becomes very important at the end of the day. The next bullet, see the PD-1 pathway. This I will need to have some time to explain. So what happens, the programmed cell death pathway? See, there are three mechanisms. Either you block, or either you put the brakes, or you do something which is very intelligent. While blocking, what would you do? See, cancer cells are so smart. Cancer cells are almost like other cells. You know what? They have their own genomic entity. They have their own morphological entity. I'm talking about genes right now, surface proteins, genes, and all that. It's a bit complex, but let me make it slightly lucid. See, they try to evade the programmed cell death Just imagine, they're so intelligent. I am giving them a chunk of T cells which will go and kill them, say engineered or non-engineered, whatever it is, but they're very smart. So what do is that they have this PD1 on their surfaces, right? So what happens? When we put, or when we infuse the cells, it will try to evade. बचने का कोशिश करेगा वो. So what happens at the end of the day? What can we do? We can engineer those cells. Whatever cells we are putting into it, the CAR-T cells, the CAR-Ts or the armored T cells, whatever it is, or the trucks, whatever it is. So what we do is that we can do three things. Number, we can block. How can we block? See, PDCD1 actually codes for PD1. So PD1 of my cell, which I'm putting into the recipient or the diseased person, will actually recognize the PD1 of the other cell, of the T cell, right? Now, what we do is that using CRISPR, we can simply cut off from my cell, from the cell that I am providing, we can simply cut off the PDCD1 gene. That's it, end of the story. So now the PD1 of my cell is never going to recognize or bind to the PD1 of the cancer cell, so the cancer cell cannot escape. This is called blocking. The next part. How do you put brakes? There's a brake system as well. So what happens in the brake system? The brake system, I push my cell, and what I do is that I engineer my cell in such a way that the PD1, though it's a less powerful part, according to me, I've gone through the literatures. Sorry for the interruption. By the way, what happens is I put my cell in such a way the PD1 expression in my cell gets downgraded. So what happens? At the end of the day, yes, they go and meet, my cell and the cancer cell, they go and meet, but the cancer cell still fails to evade because my PD1 is weaker today. It's very weak. It's a less powerful model, but the best part and the most cashy part of the entire thing, the clincher. Now, what I do, I told you that my cells are intelligent and I am also intelligent. At the same time, I engineer my cells in such a way that my cell goes with an intact PD1 now. Now there is no difference with the intact PD1 on the outer core and with a CD28, to a very specific CD28 on the inner core. So what happens? When my cell and the cancer cells meet, the cancer cell gets fooled. उसको बेवकूफ बनाना है मुझे. He gets fooled. Fine. मेरा cell के पास भी PD1 है. उनके पास भी है. They go and join, but what the CD28 inside the cell does, it goes and it do ask my PD1 cell, or my PD1 cell, of course, to behave in a different way so that it goes and kills the cancer cell. So the cancer cell gets fooled. I know I'm using a hell of personification, but that is how I will be explaining all of the part. So I am still able to fool the cancer cell. And that is why these things come into play. That's one molecular therapy there. Next one. So this is a less powerful model. In this case, sometimes, of course, we engineer the cells. It's ex vivo engineering. We engineer the cells. Sometimes we do not do. In cases, in allogenic settings, we usually go for it. And also in autologous situations, we pull up the cells from the recipient itself, and then we engineer it and put it back. It's the CAR-NK and CAR-NKT systems, natural killer cells. And CAR stands for chimerism antigen receptor. But I'll say that on the last slides, right? This is a less powerful model, but yeah, it will probably help you to give you a potent GVL reaction and a less amount of GVHD. I will not spend much on this particular slide. It's about the natural killer cells. Well, next one. Right. It's regulatory T cells and architecture of immune tolerance. Now, this is where I'm going to spend some time. See, the American Society of Hematology actually started the ORCA-T trial. The ORCA, ORCA, ORCA-T trial. And it was a very successful model rather. This ORCA-T stuff, I explain it in very lucid language. It consists of TREGS, number one. Number two, it consists of regulatory T cells. And number three, it is the conventional T. So there are three kinds of T cells. Number one, the TREGS, TREGS, the TREGS. This is a beautiful part. All are T cells. So number one is the TREG. Number two is my regulatory cell. And number three is the conventional cell. Well, let me tell you, TREGs, what do? They go and calm the environment. Calm. Ruko zara. Something like that. Ruko zara, mujhe kuch karne do. Something like this. It goes inside, my cell goes inside, my TREG goes inside the recipient. And what does it do? It will request everybody to calm down and will start to build the immune system. I will talk in details. And start to build the immune system once again of the patient. So what shall the regulatory cells do? They will go and, like a police or like a cop, they're going to go and see the scenario out there, and at the same time, they're going to send a message. GVHD घटाना है और GVL मुझे बढ़ाना है. The last part, the conventional T cells. The conventional T cells simply go and जब सब हो गया, like the first part, the TREGs, they go and calm. The regular T cells, they go and pull down the GVHD. They make a beautiful scenario, right? It's like a house that I build, and in that house, after all, my family members will go and stay. So now the cytotoxic T cells, they go, that's the third component of this ORCA-T, they go and they attack the cancer cells and kill them. Let me talk a few about TREGs. The TREG part is very important. And it's very important to understand. See, what TREGs do. Number one, TREGs have three things. What do? They go for interleukin-2 starvation. They have got a three-tier modality to act. IL-2 starvation is the first part. So what happens? These TREGs, they express a lot of CD25 on the surface. And IL-2 is extremely notorious. Extremely. So what do? They go and, they go and magically vanish off the IL-2 environment. The IL-2 chaps out there. Or the cells which produce IL-2. They go and simply vanish them off. The next part. See, TREGs also have cytotoxic T cell receptor or T cell protein. It's CTLA we call it. CTLA4 we call it actually. It's cytotoxic T cell receptor antigen or protein rather, CTL4. cytotoxic T cell receptor protein. Just beg your pardon, I have just come from some work. Right? Just CD alpha, they have the CD alpha, and what do? If my TREG has a CD alpha, they go and they deactivate the antigen presenting cell. Let's take this up in this way, in this fashion. Suppose I push my TREG with a CTL4 out there. Now once my cell is introduced, উপযাজককে বাংলা কি বলে, I don't know that. Some people, বাংলা উপযাজক বলে, I mean to say some idiots, let's call them idiots, some idiots which are known as antigen presenting cells, they automatically come and they try to create a harakiri. So what does my TREG do with a CTL4? They say then knock out those cells too, those antigen presenting cells. And the process is called phagocytosis. Right? And how do they do that? Because these APCs they have in the body, it's easy to recognize these people. These idiots, they have CD80 and CD86 in their body. So my TREG with a CDL4 goes and knocks out everything. The last part. So this is what we call the clock effect. Clock effect, I believe. Even this TREG, they also ask the system of the recipient or the diseased person, shanto jao, shanto. How do they do that? It's via throwing off all the idiots or the imbeciles or whatever it is. Let me not use harsh words. It's like I want to actually make it very lucid. So what do they do? The three things, three more culprits, which actually create much more problems. It's CD35, it's tumor necrosis factor, right? TNF gamma and CD10. Or I believe it's TNF beta, TNF, tumor growth factor. Yeah, it's TNF. It's TNF gamma. TNF gamma, CD10 and CD35. My TREG will go and tell them, just keep quiet. And in the meantime, you'll keep on flushing out these people. You'll kick them out of the system. Right? That is the ORCA T. TREG, reg cells, regulatory cells, cross, and the last part being the cytotoxic cells. They will simply go and kill. It's like, let us imagine, it's like a war situation where you have the forefront warriors. They go and they give the first hit, then comes the second, and then comes the third. Let's think the cytotoxic T cells as snipers. Once the field is clear, they will, kind of finish up the cancer cells. That's ORCA T. Well, this is what we need. Unwanted slide. Pre-transplant, we need deep cytoreduction and MRD, minimal residual activity negativity. Post-transplant, there must be a relapse prevention. ये दोबारा नहीं होना चाहिए. एक बार हटा दिया तो हटा दिया. That is it. That is our target. Post-transplant, right? Of the CAR-T, or the ex vivo CAR-T cells rather. Right. So this is another interesting part. This is another model. So even after all of this, even after all of this stuff, we still need to be more precise. That is why I have been focusing from day one, and in all of my disquisitions, my lectures, I have kept on saying that, no, you see, you need to be more and more and more precise. Let me take the example of, say, Satyajit Ray. Ray, he was one of the most colossal directors of the Bengali cinema. Okay, for Bollywood, I can help you out. It's Amir Khan. Goes for is an absolute perfectionist. Okay, for Hollywood, it's who shall I take? I'm gonna take Christopher Nolan. I'm gonna take the husband of this one, this Kate Winslet. I forgot his name. James Cameron, maybe. So you have to be very perfect at the end of the day. So what we do, we at the same time, while we are doing all of this, is ex vivo engineering, putting those cells inside in various models. I've just discussed two of them, or two to three models. See, we need to actually control this part as well, so that they do not misbehave. So in that case, the first bullet, suicide switches. That's very interesting. Now, my cell goes inside. At the same time, I see the activity of my cells, and then I inject a drug called Remedicit. Once I do that, and if I see my cells are not working properly, I will switch them off. There are suicide switches. And it's done by a drug called Remedicit. Right. Now, if I go and see that my cells are behaving, once they're behaving in a proper manner, once they are not behaving in a proper manner, can be either way. Because I have told you that all the cells are very intelligent. You know what? So if they behave in the proper manner, we use a molecule called rapamycin, which will go and push the cells, push forward the cells. Then you act more, right? But if we see that it's not behaving properly, or to be more precise, not behaving up to the mark, then we can switch it off with a drug called dasatinib. And if you see that everything is a harakiri, I've said about Remedicit, right? My first, in the suicide switch part. Same time, we see that fine, everything is proper, but still we're having this inflammation part, we're having a bone marrow suppression, we're having lots of things. My cells are not behaving properly, something of that sort. So what we do is that we depend on two more drugs, tocilizumab and dexamethasone. That is another thing of great importance. That's how we do. If my drug doesn't work, we just knock it off with Remedicit. If my drug behaves, it depends on the behavior. We use either rapamycin or dasatinib, or if we see that okay, everything is fine, but the body is behaving badly, we use tocilizumab and dexamethasone. Right. Okay. So this is another stuff where GVHD is, of course, engineered. It's not treated. And GVL is amplified. I've been speaking about it. Right. There's a take-home message down there. That's also not important. Let me come to the next one. Okay. Okay, this is very important. The CLL1 CAR-T phase 1 to first lymphodeval durable GVL. Let me tell you that right now it's on phase 4, I believe. When I first read about it, it's the most popular one. I read about it, it's on phase 4 at this point of time. Phase 3, no, not phase 4, it's on phase 3. Phase 4 के बाद तब आपका बाहर ही आ जाना है. So it's not like that. It's on phase 3 as of now. It's precision 1 trial. Now here, let me explain. So I engineer myself. Now, once again, come back to my first slide. The cancer cells are very, very, very smart. See, they also mimic the normal cells by expressing CD33 and CD123. They also behave like normal cells. This is something which actually मेरा होश उड़ा दिया था जब मैंने पढ़ा ये मॉडल के बारे में. It's very recent model on phase 3, precision one trial. So they behave, they mimic the normal cells so that they can evade and escape. So what do you do in that case? And what does CLL stands for? That's the cache. C-type lectin-like molecule. Now, see, how do they mimic? I told you, both a normal hematopoietic stem cell or any cell. Let me speak about hematopoietic stem cell. I have to be a little more precise now. A very normal hematopoietic stem cell in a diseased person expresses CD33 and CD123, and there, these chaps, the cancer cells, even they produce the same thing. They show the same thing: CD33, CD123. It's one hell of a problem. So what do? We found out, or the scientists did find out, must give all the credit to the scientists. Well, it's not me sitting out here and lecturing you. It's not like that. CLL1. This is specifically expressed by the cancer cells. The C-type leptin-like molecule, or leptin-like molecule, yes. They are expressed more by the cancer cells. The hematopoietic stem cells, yes, they do. They also do have CLL1, but in a very, very less proportion. Suppose a cancer cell will have 100. A normal cell will have 1 or maybe 0.5. So now we can go and attack the cancer cells easily by recognizing the CLL1 domain. Sounds good, and you can see the overall survival rate of the patient, it goes up to 12 months. And almost for 2 years, it goes up to 50%. 2-year overall survival rate. And also we have a subset which has a minimal residual activity. That is what this model claims of. You can go through the net, you can check them on LinkedIn, you can check them anywhere else. But this is how it is. On the NLM catalog, right? So this is one of the first platforms suggesting a true transplant-like immune surveillance. So the next slide. So you see, why does this out from the others? These are the trials. CD123 CAR-T, UCART123, CD7 CAR-T, dual CARs. I spoke about ARMA-TRI, spoke about TREGS. I spoke about NKs. I did speak about interleukins, IL-18, IL-18, 12, and all those other things. PD1 rather. I spoke about the ORCA team. But this is one part which is the best. See the complete remission rate, CR rate, complete remission or cure is very, very high. So dhire dhire we are approaching to that part where we can start, we shall start, say 10 years from now on, we shall start curing patients. Durability, of course, the best one so far, and verdict is most balanced. There's no niche targeting, it's promising, it's limited, future strategy. As of now, this is the most hefty part, the CLL1 CAR-T. That's it. The pleasure, colleagues. I spoke about 5 models, I believe. Just go back to one slide, I believe. One particular slide. I did speak about this part, this one, the NK cells. Where is the natural killer cell? Where are my natural killer cells? Yeah. Let me go to this natural killer part. In this part, of course, we can pull out the cells and go for ex vivo engineering of the cells, make them slightly a bit more intelligent, push them in the body for a precision medicine with a very for precision medicine with a very good outcome, for a fantastic outcome, or maybe the patient can be cured as well. Particularly, why I have come back to this slide is this CAR-NK or CAR-NKT system, they can be used in several infections as well, such as HIV, Hep B, in some inflammatory conditions like SLE, myositis, and so on and so forth, or systemic sclerosis. You've heard about the name, maybe. It's one of the most dangerous diseases, right? So this is where this thing can actually help you. The NK part, natural killer part, this model. You can use it for other diseases as well. And you can see the source. If you have any queries, you can just mail me on my mail ID, drbiswasmd@aol.com. This is one of the most highest in topics, and I've actually made a very latent attempt. I don't know if it was up to the mark or not. It's up to the verdict of the listeners, those who are out there listening to this chap out here. Well, this is the chap straight away, and I thank Medvantage for giving me this opportunity to speak on this topic. Research, as far as I see, it's still on the way. And every day I find something new, say on LinkedIn, say on ResearchGate. There's so many parts of it. But yes, maybe it's like hum honge kamiyab at one point of time. Yes, maybe we'll try to kick it off. Kick off those cancer cells. As doctors, we should bring back the smile in the face of those who are deceased on humanitarian grounds. That's the catch, and thank you, Sanjay sir. And Medvantage will provide you more solutions. Check on the links of Medvantage. I'm pursuing a course right now on critical care medicine. I must say, it's ongoing. I'll have a hands-on after this, after my theories are over. After that, I'm going to get a certificate which shall be accredited by NHS, England, RCP, CPDE, integrity, and things. So I've opted for this and encourage everyone to opt for Medvantage. Thank you, and to the pleasure, ladies, gentlemen, docs, my fellow docs, seniors, juniors, and whosoever were there in this disquisition, but never forget the Boolean logic. Another clean ship, and my last statement. Thank you, and to the pleasure. Any questions from any side? Any questions? Any questions? I'll be happy to answer. And if anybody would propose me a better model, because I've gone through plenty of models, I thought of speaking about 5. So, right. Any questions from any side? No questions. It's really strange. I'm also having a lot of questions as to how this guy ended up speaking about so many things. Any questions from any side? Well, if there are no questions, right? Sir, can I? Sir? Hello?
Yeah, sir. Thank you so
We
much
can end it then. In
for the
that
session.
case, I hope you like the topic as well.
Yeah, sir.
So this is how it is, and thank you so much.
Thank you, sir.
To the pleasure, sir.
Okay, thank you, sir.
Right away, right away. Thank you so much, sir. Thank you.